So our next speaker, I'm going to come on down, is Safina Taitano. She is a PhD candidate in the Department of Immunology, and she's going to discuss the roles of IL-4 and IL-5 in our B regulatory cells.
Good afternoon, everyone. Thank you for staying for my talk. Today, I will be discussing some interesting data that we found for the role of IL-5 in stimulating B regulatory cells and the antagonistic role of IL-4 in this stimulation. So autoreactive B cells can be activated by autoantigens or self-antigens, and this can lead to the differentiation into pro-inflammatory B cells. These B cells can interact with autoreactive T cells, and this interaction can enhance the activation of both the cells, inducing pro-inflammatory cytokine release such as IL-6, IL-17, and interferon gamma, along with the secretion of autoantibodies. Now, this is what is normally thought of autoreactive B cells. However, our lab is interested in the regulatory functions that they can have, such as the expression of the death-inducing ligand, Fas ligand, and the secretion of anti-inflammatory cytokines such as IL-10. And so these killer or regulatory B cells, which we believe to be a complete separate subset from these normal B cells that you would think of in your autoimmunity, these B cells are thought to interact also with autoreactive T cells in two ways. So first, through the secretion of IL-10, and this inhibits the expression or production of pro-inflammatory cytokines, and it also induces the induction of T regulatory cells, which themselves then go on to express IL-10. The second being through Fas ligand, which induces apoptosis in the autoreactive T cells, eliminating them from circulation. And so both of these mechanisms may lead to protection from autoimmunity and therefore may be potential therapeutic targets. Okay. So many of the studies looking at B regulatory cells use B cell mitogens to activate them. And so we wanted to look at a more T cell derived stimuli. Previously in the lab, we found that activation or stimulation using the CD4 ligand transduced fibroblasts and IL-5 increased the killing ability of Fas ligand positive B cells and the secretion of IL-10. Since IL-4 and IL-5 are usually cooperative cytokines, we also wanted to look at the effect of IL-4. And interestingly, we found that IL-4 actually inhibited the Fas ligand killing ability of these cells. So now we want to determine what the effect of IL-4 is on IL-10 secretion. And this is what this talk is mainly focused on. So for the rest of the experiments that I'll be showing you, we're going to be using the same culture setup here with the irradiated CD4 ligand expressing fibroblasts and different cytokine conditions, either IL-5 and IL-4 alone, together, or no cytokines at all. So initially, we wanted to determine or confirm that this wasn't a strain-specific phenotype. And so here we're using DBA, BLAC6, and BALB/c mice splenic B cells. And we see that in all three of these strains, IL-5 significantly increases the production of IL-10. So this is not strain-specific. And additionally, since we found that the Fas ligand positive B cells were enriched in the CD5 population, we also wanted to determine if the IL-10 producing B cells were also enriched in this subset. So here in the purple bars, we're going to be using splenic B cells that are FACS sorted to be CD5 negative. In the orange bars are going to be total splenic B cells, and in the blue bars are total splenic B cells from mice that are deficient in CD5 positive B cells. So here in the IL-5 culture condition, you can see that in the absence of CD5 positive B cells, you no longer get this significant increase in IL-10 production. So CD5 positive B cells are the dominant producers of IL-10 in our culture. Now when we look at the effects of IL-4, we see that when IL-4 is in the culture alone, it does not induce IL-10 production. But more interestingly is that when it's combined with IL-5, it's actually blocking this increase in IL-10 production. So to look at how potent the effects of IL-4 are, we did a dose response. And so we see when there's no IL-4 in the culture and there is IL-5, again, we have this elevated levels of IL-10. However, when we have just 5 nanograms per mil of IL-4 with 80 nanograms per mil of IL-5, you already see a very significant reduction in IL-10 production, which is maintained as you add more IL-4. So the IL-4 has a very potent effect on blocking IL-5 action. Okay. To look at the kinetics of IL-5, we also did a time course, and we found that IL-5 starts to significantly increase IL-10 production around 72 hours, while none of the other culture conditions increase IL-10, as previously seen. And since IL-10 is an anti-inflammatory cytokine, we also wanted to look at a pro-inflammatory cytokine. So we looked at IL-6, and we found that IL-4, either alone or in combination with IL-5, significantly increases the IL-6 production compared to IL-5 cultures or no cytokine cultures. And this is evident as early as 24 hours. To look more closely at the mechanism of how IL-4 is affecting these different cytokines, we decided to focus on STAT6 since it's the dominant transcription factor that IL-4 normally signals through in other cell types. And so here we're going to be using splenic B cells from BALB/c mice or STAT6 knockout mice. And so we see that here, in the absence of STAT6, IL-4 is no longer able to block the secretion of IL-10. Additionally, IL-4 is no longer able to induce IL-6 either alone or when in combination with IL-5 in the absence of STAT6. So IL-4 is signaling through STAT6 to mediate these effects. So that's all the mirroring studies that I will be showing you today. However, we were able to do some studies using B cells from human blood from healthy donors. So this is the same culture setup, and we decided to do a time course study, which really almost exactly mirrors the mirroring study. However, the important difference here is that in the human B cells, IL-5 was not necessary to induce IL-10 production. And so in the blue here, we have the no cytokine condition, and in the red, we have the IL-4. And you see that in the no cytokine condition, you start to get IL-10 increase around 72 hours. And again, IL-4 increases IL-6 production as early as 24 hours. Okay. And so this is basically the same data, but we wanted to look at it at an individual level. And so each line here is an individual person. And so we see that IL-4 consistently inhibits IL-10 production and consistently increases IL-6 production. So in conclusion, CD40 ligand and IL-5 stimulate B cells to secrete IL-10, and this is potently inhibited when IL-4 is present. IL-4 also induces IL-6 secretion in mice and in humans, and in mice, this IL-4 effect is mediated through STAT6. In the future, we want to look deeper into the mechanism of how IL-4 is affecting these cytokines. And we also want to investigate in vivo situations that may mimic our cytokine environments, which would be helpful in studying these cells and their function in the immune system. Lastly, we're hoping to determine if these cells have therapeutic potential. I would like to thank my mentors, Steve Lundy, Nick Lucas, and David Fox, and also the Moore Lab, which is where I do most of my work, also in the Fox Lab, and my immunology program, and of course my funding. Thank you very much.
We have time for a couple questions. Yes.
Hi, nice talk. So I noticed in your results when you were doing the dose-dependent changes in IL-10 concentration in the mouse studies, that one, that it looked like you had an increase with just a low dose of IL-4 with no IL-5. Was that something you were consistently finding? And what dose of IL-4 were you using throughout the rest of these experiments?
So the dose of IL-4 that we're using for the rest of the experiments is 50 nanograms per mil. And in the IL-4, IL-5 combination cultures, those are going to be equal, 1-to-1. Are we concerned about this? I don't think this is significantly elevated statistically, so we're not really concerned about that. And since, yeah, that's all I can say. Thank you.